Welcome back. It's a pleasure to introduce our next speaker, Dr. Bill Andrews. Dr. Andrews is the founder and CEO of Sierra Sciences, a company focused in ways to extend human healthy lifespan and lifespan, telomerase maintenance. Telomeres are found on the tips of the chromosomes and have been shown in thousands of scientific to be the clocks of aging in humans. When telomeres get shorter, we get older and our health declines. As a scientist, athlete, and executive, Dr. Andrews continuously pushes the envelope and challenges convention. He has been featured in Popular Science, The Today Show, and numerous other documentaries. Bill has been a medical researcher in biotech since 1981, focusing on cancer, heart disease, and inflammation research, though his passion has always been aging. In the early to mid-1990s, while at Geron Corporation, Dr. Andrews led the research to discover both the RNA and protein components of the human enzyme called telomerase. This enzyme is responsible for preventing telomeres from shortening in human reproductive cells, and this is why our children are born younger and we are even thought they come from our old cells. Inducing this enzyme to lengthen telomeres in all our cells, not just our reproductive cells, to reverse aging and declining health due to aging, is the principal goal of Sierra Sciences. Dr. Andrews was awarded second place as National Inventor of the Year in 1997 for his cancer research. Also earned his PhD in molecular and population genetics at the University of Georgia in 1981, and then served as senior scientist at Armus Corporation and Codan Corporation, Director of Molecular Biology at Berlex Biosciences, and also at Geron Corporation, and Director of Technology Development at EOS Biosciences. All of this before starting Sierra Sciences in 1999, where he now serves as President and CEO, as well as Chief Scientific Officer. Dr. Andrews is also inventor of over 50 plus, has more than 50 US issued patents on telomerase, and is also the author of numerous scientific research studies published in peer-reviewed scientific journals. Dr. Andrews, thank you so much for taking the time for being with us. It's great to have you. The stage is yours now.
Thank you very much. I have really been enjoying this conference so far, a lot of very excellent speakers. I am very honored to be invited back to speak again. I'm going to begin my presentation now, but I want to start off with something that I think is truly amazing. Remarkable things have happened in aging. I used to get excited about things like Benjamin Franklin living to be 84 years old. But now we have a 90-year-old going into space. And I think I wasn't as impressed with the fact that he went into space as much as I was that he climbed 7 flights of stairs just to get to the capsule. So I want to say congratulations, William Shatner. You're a poster child for everything all of us want to be. And I can only say we've come a long way in terms of aging if we look back as far as Socrates and Benjamin Franklin, people that are known to have lived a long time, but William Shatner has blown them all over. I've spoken at several, this is my 4th presentation at Healthy Masters. And I don't like repeating myself, so I'm going to just quickly go through what my other presentations were about, and you can go back and look at those. The first one was December 1st, 2019. In those, I just spoke about telomere biology in general and gene therapy. Then again, in November 29th, 2020, I spoke about what are called go-to-market partnership opportunities, a way that we are generating funding for my research. I also talked about myself, my history, my accomplishments, my background, everything that made me what I am doing right now. And then again, I spent a little bit of time talking about telomere biology again. Then on May 30, just a few months ago, I spoke again, and this time I just talked about my beliefs in general about aging. I talked and aging and other things like COVID. I talked about funding strategies, and again, I talked a little bit about go-to-market partnerships. So if you want to hear about that, because I'm not going to be talking about these things today. People usually expect me to be talking mostly about telomere, but I'm not today. Today, I want to talk about aging just in general, the basics of aging, and also, more importantly, what we're doing to curing aging. And when I say curing aging, I mean anything related to slowing it down, stopping it, or reversing it. And I want to say I've been obsessed with this my entire life, but most of my life, everything I read or heard about aging didn't really make sense. And that was until I met Mike West. And Mike West taught me a lot about aging and steered me on the right path. That's the reason I went to Geron Corporation, was to work with him. And I was very excited to see that he spoke yesterday. And I can tell you that everything I'm doing is still in following the teachings that he has provided me. I also want to say there's a lot of misinformation about aging and curing aging. And not just aging, but everything. There's COVID. We're experiencing more than ever before with all the misinformation that's being spread about COVID. Instead of like COVID, should we wear masks? Shouldn't we wear masks? Things like that. But on the subject of just aging, I want to say first, aging has not been cured yet. Okay, contrary to popular belief. There's a lot of people saying they've reversed aging, they've cured aging, stuff, but we haven't cured aging yet. Otherwise, William Shatner would look like the picture on the left, and he doesn't. Okay, and that's what we've got to do. We haven't cured aging until we can make people back to what they were looking like, feeling like, and behaving like when they were 25. I think William Shatner was actually older than 25 in that picture. So what I want to do is I want to spend a little bit of time talking about what to believe and what not to believe, only because there's a lot of misunderstandings relatively in aging and COVID. So I do speak a lot about this in other presentations. You can go to my website, you can see much longer versions, but I just want to go briefly over the basics. One is, we have a lot of people that are just trying to first promote themselves, promote their companies, promote their products. And so there's a lot of misinformation. And it's not just the speakers that are a problem, it's the audience too. The audience tends to believe those things which they wish to be true. And for instance, masks. If somebody says you don't need to wear a mask, people generally readily believe it when it comes to COVID. But the thing is, you've got to really go look at the data. The other thing that people, they tend to believe entertainers more than they believe real scientists. And then again, a lot of people get on the news and things like that and call themselves real scientists when they really aren't really scientists. They're spreading the words that they've heard from real scientists. And often they get it wrong, and I'll come back to that. But one is look at the data, not just somebody's interpretation of the data. An example of data is, somebody does some study, they find that 40 people are in group A and 60 people are in group B. And when I say groups, I mean the results, okay? The results of the study show that 40 are in group A, 60 are in group B. And somebody's interpretation of the data is to say that 40% of the people are in group A. I mean, it's like a lot of times people don't do the calculations correct themselves. They're not really good at data analysis. Now, a lot of people, scientists, aren't really trained in data analysis. They're trained in actually doing technical work at the lab bench. So be very careful. If you read a paper and they don't show the data, question it, okay? So one is believe the data, but this is only the beginning. So don't look at the interpretation of data, look at the data. Two is focus on peer-reviewed studies. I'm going to get to aging in a second. I just want to briefly get over this stuff about how to believe what's true and what's not true. But read peer-reviewed studies. A lot of studies get published, but nobody peer-reviewed them. I mean, books, documentaries, things on the news, newspapers. Nobody peer-reviews these. People just, a lot of times things get in the news just because the newspaper or the TV station think it's going to help with ratings, okay? Because somebody's saying something that's very interesting or that people are going to be listening to, but it's not peer reviewed. So look at peer reviewed studies. There's going to be a recording. I don't want anybody to write these down, but these are two good links to websites where you can find peer-reviewed studies, databases of peer-reviewed studies. Not just in medicine and aging, but also in politics. For instance, the second one, everything under the sun. So you can actually find peer-reviewed studies on just about every subject there is going to those two links. Now, looking at peer-reviewed studies can be a problem because even peer-reviewed studies sometimes slip through the cracks. Sometimes you can find two peer-reviewed studies that say the exact opposite. Well, both of them can't be right. So what I also encourage is to do what's called meta-analysis peer-reviewed studies. And that's where you actually look at all the studies. And I'm just going to show an example here. You look at a bunch of studies on the same subject, and you find that two of the studies conclude that conclusion A is true. 16 of the studies conclude that conclusion B is true. Then you're going to find some of them give no conclusion. And then, believe it or not, a lot of times some papers actually come up with both conclusions are true. Okay, but the bottom line is by doing meta-analysis, you are not focused on those studies that are saying what you wish to be true, which is oftentimes why conclusion A gets accepted, because it says something people wish to be true. Doing this, you find out that conclusion B is the more likely one to be true. Now, you can carry this one step further if you're really good at data analysis, experimental design, statistical theory, logic, all that kind of stuff. You can do what's called critical meta-analysis of peer-reviewed studies. I mean, this requires a lot of work if somebody's really passionate about really knowing what's true and what's not true. I highly recommend you get really good at doing things like this. But in this case, you read all these papers, but then you look to see, well, was their experimental design correct? Did they analyze their data correct? Did they do their statistics correct? Did they interpret the data correct? All this kind of stuff. And so when you do this, you find, actually, conclusion A, both papers really were terrible. They did not. And that's probably why they came up with an opposite conclusion as B. But you might find that some of even conclusion B papers actually did it wrong. And you find out that only 13 of them really made sense. You find out none of the ones with no conclusion made any sense, and none of the ones that said both A and B made any sense. Well, again, you come up with conclusion B, but now you're more certain of it. Okay, and so this is really where people need to get more involved. Don't quit believing the things you wish to be true. Quit just listening to entertainers. Quit believing people that just say they're the experts, top experts in the field when they're typically not. And so anything else you hear from anything besides peer-reviewed studies is just hearsay. Okay, now I don't want to say don't be a victim of hearsay, but it's kind of like the telephone game. Remember the telephone game? You whisper in something in one ear and somebody whispers that same thing in somebody else's ear. When it finally gets to the last person's got something totally misunderstood or repeats it exactly incorrectly. Well, the same thing is true in our corporations. Our corporations and our big industries like the FDA, the WHO, the CDC. I mean, we are hearing from the people at the top. They're the top experts, but they don't have time to be doing the studies themselves. They're not doing this. It's been years and years since they've done any studies. They are actually just getting this information from the bottom where the scientists are. If you really want to know what's real and what's not real, go look at the scientific publications that these scientists did. Okay, and that's the best place to get it. So again, don't be a victim of hearsay. Go to the peer-reviewed studies. Don't listen to people just because they say they're top experts. Okay, so that said, here's what I want to say about aging from my perspective of doing critical meta-analysis of peer-reviewed studies. And I want to compare the search for aging as search for the source of the knowledge. And the reason why I like this analogy is because a lot of people ask me, how long is it going to take to cure aging? Or what is it cause of aging? And things like that. And it's when these two scientists, doctors, were first searching for the source of the knot, and somebody asked them, how long is it going to take you? They didn't know the answer to it because there were too many forks in the river, there were too many dead ends, there were too many problems. There was no way they could tell how long it was going to take for them to find the source of the Nile. So I want to change it. This is in search of the sources of aging. And I say more than one source because aging is probably multiple things are causing aging in humans, and those same things causing aging in humans are probably not the same things that are causing aging in other animals. But the 4 different subjects that I want to talk about is, one is what is aging, why we age, causes us to age, and how we can treat aging. And so I want to say first, nobody really knows. Okay, you do the critical meta-analysis of scientific peer-reviewed studies, and you find out that it's still all guesswork. Nobody really knows the answer to any of these things yet. We're figuring it out, but maybe we're going to have to figure out how to cure aging before we can figure out what aging is. So I'm going to call these Bill's best guesses, okay, because of my critical meta-analysis, and I've put everything together. I can't say anything's for certain. I can just say, here's my best guesses based on all the studies that I've done. And so first, let's talk about what is aging. Well, as we all know, there's a lot of biomarkers of aging now. There's telomere length measurements, there's DNA methylation, there's glycosylation measurements, and these are very important things. I think they're a good measure of health. I think you want your methylation patterns to be ideal. You want your glycosylation patterns to be ideal. You want your telomere lengths to be at the right length. But does that really say anything really about aging in general? Well, the bottom line is we all know which photo of Betty White was taken first. And so we do know what aging is, even without biomarkers. It's just really hard to put our finger on exactly what the difference is. Yes, we can say there's more wrinkles in the picture on the right, her hair color is different on the right, but there's a lot more going on there. And it's not just appearance, it's also how they behave, how they feel. Okay, so I call that aging. Aging, whatever, it's not, I call it qualitatively quantitative. At Sierra Sciences, you'll hear us use a word all the time called the QQ score, which is called qualitatively quantitative, which means we can't really put a measure on it, but we can say one is different from another, okay? And we can repeat that over and over again. Well, we all can tell which picture of Betty White was taken first, and that's all the measurement we really need to know. We're also going to want to look at, of course, her athletic abilities and talk to her how she feels. But if we can reverse that, okay, make her feel and behave and look like she did before, that's what I call the cure for aging, and I call that the Betty White test. And in honor of William Shatner, I really think that what William Shatner just did is one of the greatest things that ever happened. I am going to, in the future, be calling this the William Shatner test. And so I'm very excited about this. And so this is my introduction to the William Shatner test. Sorry, Betty White, I wish you all the best. And I'm going to now use the William Shatner test. So let me move on to why we age. This is a very controversial subject. But bottom line is, a successful species is one that can survive a rapidly changing environment. Now, variation is important in a species. And I want to say first, evolution is not a force, it's a result. Okay, so let's not talk about forces that are driving us towards a certain direction. Now, I strongly believe in something called antagonistic pleiotropy, which is a major cause of evolution. But as long as we don't start saying that what's bad for an individual in a species or a member of a species is also good for that member of the species, okay? What antagonistic pleiotropy means, what is bad for the member of the species is actually good for the species as a whole, okay? And that's something we got to remember. And we got to remember that when environment changes rapidly or even not rapidly, there has to be variation within the species. If every one of us and every one of any species was 100% identical to each other, and the environment changed, if one of those members die, all those members are going to die, okay, because there's no variation. So in order for a species to survive a rapidly changing environment, there needs to be some variation within the species. And like it or not, aging and cancer are things that are important, and I'll come back to that in a second. So the way to increase variation in a species is to shuffle our genes. Okay, the more a species shuffles its genes, the more variation it's going to have, and the more likely it's going to be able to survive a rapidly changing environment. And so, how do we shuffle our genes? And that's through mating between species. Genes, two species, member A, member B, they breed together, and they create an offspring that has an entirely different shuffling of genes that probably ever existed before on this planet. And it turns out that more shuffling is going to occur if the offspring are allowed to interbreed more than the parents, more than if the parents are allowed to rebreed. And I talk about this in a lot of my other presentations. You can go to my website, you can see me go into more detail about this. But the fact is, we're going to have more variation if we don't allow the parents to rebreed. We let the offspring rebreed, and their offspring are going to have more variation than ever existed before on the planet. So we have to do this. And so a species that eliminates its old one way or another is going to have more gene shuffling, because that's the way to improve the chances that the offspring interbreed more than the parents rebreed. So eliminating the old is more likely to allow us to survive and not become extinct. And that's why I said before, aging and cancer are important for survival of the species. So a lot of people say we age to prevent cancer, which is an example, they say, of an antagonistic pleiotropy. I don't believe that's true. I believe that cancer is a good way of eliminating the old, like it or not. But the good news is we are more experienced now. We can control our own evolution. And so we don't need to rely on just regular evolution to help the survival of our species. But all of what I just said, this is why we age, in my opinion. This is why we age. There's no proof of this, but based on the meta-analysis of peer-reviewed studies, this is where I conclude that we age just to eliminate the old. Aging is important for survival of the species. Next is what causes us to age. Well, there's a lot of causes of aging, or let's just say fewer than other things I'm going to show. Environment is a major cause of aging, mostly oxidative stress and inflammation. Genetics, some people have better genetics than others. And then there's telomere shortening. And I'm going to come back to that. I'm not going to spend a lot of time, not any time talking about telomere shortening, but that is one of the things that I strongly believe, and I'll explain why in a few minutes. But the causes of aging cause the symptoms of aging. And so a lot of things that you hear about are addressing the symptoms of aging, not the causes of aging. Examples of the symptoms, symptoms of aging are also causes of aging, okay, as you'll see, and you've heard from others speak. Cell senescence is an example of symptoms of aging. We have cell senescence because of our aging process. We also have mitochondrial dysfunction because of aging. And now cell senescence and mitochondrial dysfunction also cause aging, which is I don't want to pretend like I'm not saying that. Then there's altered gene expression and a host of other things: DNA damage, stem cell depletion, protein glycation, junk accumulation, lots of things. You've heard this from many other speakers. And all these result in age-related decline in health, decline in appearance, and death. And those are the key things that we really want to avoid. Okay, I want to focus on these things in the red square. All these things cause our cells to be unhealthy. Okay, so this is where I run into a dilemma when I think about the causes of aging. And what I want to do is I want to talk about a property of cell division. And I'm going to use bacteria as an example. Property of cell division is true for all cells, you would think, at least until 1960s, we used to think it was all cells. But if you take bacteria, and I'm just going to use bacteria as an example, you put bacteria into a petri dish, and you mix that with an equal number of unhealthy bacteria, what you're going to find, because these unhealthy bacteria are going to grow slower, you're going to find after a while that after they grow for a while, that the healthy bacteria will far outnumber the unhealthy bacteria. To let them grow further, you're going to find out that the healthy bacteria completely take over that culture. Well, why isn't this happening in humans? Why is it that we are accumulating these unhealthy cells and the healthy cells aren't replacing these? And this has been something that's been a mystery for me for a long, long, long time until I met Mike West. Mike West showed me a path that explained all this. And that's because human cells don't divide indefinitely. Leonard Hayflick showed this in the early 1960s, I think it was 1961, that for some reason human cells don't divide indefinitely, and that's why the unhealthy cells can remain in the body and actually become a major component of the body. And the reason for this is because of telomere shortening. Telomere shortening is the reason why our cells quit dividing after a while, and we can no longer replace the unhealthy cells. So, and that's our main focus at Sierra Sciences, to solve that problem. Okay, so the next question I want to get into is how can we treat aging? And there's a lot of things. We can minimize the impacts of the environment. We can regulate gene expression. We can do gene therapy. We can correct cellular and tissue damage. We can do gene editing, stem cells, cryonics, whole brain emulation, which is making our brains transfer to the cryonics and whole brain emulation I consider last resorts. Cryonics is, of course, where you freeze the body and wait, thaw the body later, maybe 100 years later when there's a cure for your disease or cure for aging. Whole brain emulation is where you transfer all your thoughts and all your brain to a computer, and it stores there until you have another body to put them into. Okay, and then telomere maintenance, which is the main thing that we're focusing on now, because we're not resorting to the last resort first. Okay, so this is the things in the square here. These are all the things that Sierra Sciences is working on. I'm very glad that many other scientists are working on many of the other things. So how do we know when we get there? Remember, this is what I was saying, that we're on the search for the sources of aging. How do we know when we actually figure out how to cure aging? Well, one of the big problems with aging research is we have no good way to measure the slowing of aging. Okay, you would have to take essentially thousands of identical twins that have been their entire life grown up exactly the same, and then you would have to divide them into two groups, one twin in one group, one twin in the other group, treat one twin differently than the other, and see if this slows down their aging. After maybe 10 years, you could see a difference in aging. That's impractical, and so we have no really good way to measure the slowing of aging. We have the biomarkers, but as I said before, it's not clear that the biomarkers are really good measures of aging. But they are good measures of health. So to really know that we've been successful at curing aging or treating aging, we need to show that we can reverse it. Because as I said before, in the William Shatner slides or the Betty White slides, we know without a doubt which photo of Betty White was taken first, which photo William Shatner was taken first, and we can measure using that qualitatively quantitative QQ measurement. So when we do this, then and only then will we know that we have that our search for the sources of aging is complete. And with that, I want to remind everybody that I do have other presentations. If you want to hear more about gene therapy and my background and who I am, they'll go to those presentations. And I want to thank you very much. And I especially want to thank Mike West. I was very excited to hear him speak yesterday. I want to thank Mike West for showing me the correct path to follow. Thank you.